All right, in this video lecture, I am covering 13.3 DNA replication. And so I'm going to talk about how DNA copies itself. So I'm going to we're going to talk about specific enzymes that do specific jobs, and then compare DNA replication in prokaryotic cells, cells that do not have a nucleus, versus eukaryotic cells, cells that do have a nucleus. So let's just quickly review features of DNA. All right, so the sugar phosphate backbone, I believe, is what we are looking here. Hopefully I don't have a word that's faded in. So this is our sugar phosphate backbone. Here's our nitrogen bases. And I think that's just a plain old nucleotide. Okay. DNA is considered a polymer. Okay, what macromolecule does it belong to? That would be nucleotides. What is DNA's monomer? What are its tiny little subunits? That would be a nucleotide. What is a nucleotide made up of? It's made up of three things. Okay, sugar, phosphate, and a nitrogen base. What are the four types of nucleotides? Adenine, cytosine, thymine, guanine. What is the base pairing rule? A pairs with T and C pairs with G. Who got credit for the structure of DNA? That would be Watson and Crick. All right, so getting on to the notes here. DNA replication is all about copying that code, so that way the new cell has the same instructions to carry out functions for that cell. So replication is just when DNA is duplicated or copied, and it occurs in interphase, specifically the S phase, right here, this green arrow. So we discussed the cell cycle, G1, S, G2, and then mitosis with cytokinesis. And so a single strand of DNA can act as a template due to our base pairing rules, where A pairs with T and C pairs with G. So if you have one half of a DNA molecule, you can construct the other half because of the base pairing rules that exist. In order for DNA to replicate or copy itself, it needs some enzymes to carry out the work. So DNA, is just an information molecule. It's proteins that actually do the work. So they carry out the process of replication, not DNA. So lots of different enzymes we need to be aware of. First off, DNA helicase is an enzyme that unzips DNA. So DNA is a double helix or a ladder, or a zipper, and it's got to be unzipped. Okay. So it literally breaks those hydrogen bonds and unzips it. And then we have an enzyme called DNA polymerase that joins nucleotides together to make complementary strands of DNA. So there are these free-floating nucleotides in the nucleus, and DNA polymerase matches those free nucleotides to a template or a side of DNA that's it's reading to make the new half. Okay, DNA polymerase also proofreads DNA, make sure that the nucleotides are matched up correctly, and if not, swap out the correct nucleotide for the right one. Another enzyme that you need to be aware of is telomerase, which means that it adds short, repeated DNA sequences to the ends of chromosomes. And so telomerase, anything that ends in ASE is an enzyme. And you find telomerase working at the ends of chromosomes called telomeres. Now, every time a cell is copied, your chromosomes get a little bit shorter. So I'm going to talk a little bit about that, I think, coming up here. Well, how does the replication process work? So 3 very general steps. DNA is unzipped by DNA helicase at several places, and each strand is exposed. So this is where DNA polymerase comes in and reads that templates, takes these free-floating nucleotides and pairs them complementary. It's very smooth. It happens in one direction and is a little bit more complex in the other direction. So remember, DNA is anti-parallel. It has directionality. So I'll show you a diagram what I mean by that. And then the end result is 2 identical molecules of DNA that are semi-conservative, where one strand was kind of the original strand, the parent strand, if that's how you want to think of it, and the other is a new strand that was built by DNA polymerase. So this is the diagram showing DNA replication. So let me just orientate yourself. Here's my chromosome. Uncoil it. Here's my DNA. Okay. First off, DNA has to be unzipped. So you can see here DNA helicase acting like a wedge, unzipping the DNA, so these strands are exposed. Second step, DNA polymerase binds, okay, and is reading complementary. Now, because DNA has directionality, one side of the strand is called the lead-in strand because DNA polymerase can just follow DNA helicase, okay? But the other side is called the lag-in strand because it can bind to where DNA helicase, is, but it has to go this way because of directionality. And then as it's moving this way, DNA helicase continues to unzip. So now we have all these nucleotides exposed. And when DNA polymerase meets up with a double strand, then it will fall off and then go find DNA helicase again and then match complementary. So it's pieced together segment by segment. So I have this model here. Okay, it's not perfect. Wow, it's going to be kind of glitchy. So the first step is that DNA is unzipped. Right, so I'm going to do this to show that it's being unzipped by the enzyme. Which enzyme? DNA helicase. Okay, and let's just say that this top half is my lead-in strand. Okay, so what am I going to do here? I'm going to start on this end and just follow DNA helicase. So C pairs with G, and I'm already regretting going at an angle because that means now I have to fix or tilt my nucleotides. So A pairs with T, A pairs with T, A pairs with T, G pairs with C, and T pairs with A. And then G pairs with C. Okay. All right. And so DNA polymerase would just continue on, all right, until it's done copying DNA. Now, as that's going on, we have another DNA polymerase going in this direction, all right? So I'm going to use the top half here. So C pairs with G, A pairs with T, G pairs with C, A pairs with T. Okay. Now, if I start right here and I start to compare, okay, I go CG. Well, hey, look, CG, AT, AT, GC, GC, TA, TA. And if I didn't cut it off, there'd be another TA, TA, AT, AT, and then the CG's cut off. And you can see that I have two identical DNA strands, and these strands are semi-conservative. This black sugar phosphate backbone was the parent strand. The new strand that was made by DNA polymerase, okay, that's the new strand, and I denoted it in purple here. Now, this process of copying DNA is fast and accurate, about 50 nucleotides a second at several origin spots of replication, because it happens at multiple points. I'm going to see if I have a picture. I can't believe I don't have a picture of this. It's got built-in proofreading function to fix those mistakes. That's what DNA polymerase does, and the replication happens all the time. So DNA is unzipped, DNA polymerase matches complementary, and the two new strands are identical and semi-conservative. Here's a diagram showing DNA polymerase where it will repair or fix mistakes and then continue on. Now, the final learning objective is comparing prokaryotic DNA replication to eukaryotic replication. So in prokaryotics, we only got one chromosome and it's circular. Okay, no nucleus, it's in the cytoplasm. And that replication starts at one point. So I'm going to draw a picture. Let's say this is my circular chromosome. I'm going to use purple. So it's got an origin, one spot. Okay, starts at a single point. And DNA polymerase will go this way, and another DNA polymerase will go that way, and they'll meet, and then you end up with two strands that are identical to each other. Eukaryotic DNA replication is a little bit more complex because we have multiple linear chromosomes. We're also in a nucleus. Chromosomes are larger, more complex, and so that replication will begin in several places. I still can't believe I don't have a diagram. So if this is my chromosome, okay, and let's say that I have a spot here, that replication starts, and replication starts here, and replication starts here. So then pretty soon I see something like this. And we'll say the new strand is black. Okay, that was really bad. But it starts at several places, and they will meet and fuse together, and then you end up with two identical copies of that chromosome. All right, so that does it for 13.3 DNA replication.